Welcome back, guys. We'll discuss today's sperm transport. The basic objectives of today's discussion will be covering the basic anatomy of the sperm and the male reproductive tract, the transport of sperm through cervical mucus, uterus, uterotubal junction, and advancement of sperm beyond the tubal reservoir. And then finally, the exits of sperm toward the oocytes. We will be covering all these topics today. So, sperm is a male gamete and is derived from Greek word sperma, which means seed. Head of the sperm contains a membrane-bound nucleus capped by acrosome. Inner acrosomal membrane closely opposed to nuclear membrane. The outer acrosomal membrane next to surface plasma membrane. Flagellum is a complex structure of microtubules and fibers surrounded at proximal end by mitochondria. Motility and ability to fertilize are acquired gradually as the sperm passes into epididymis. So this point is very important. When the sperm passes into epididymis, only after that it acquires the ability to fertilize. Sperms are terminally differentiated cells. They are deprived of active transcription and translation apparatus. They must survive in female without benefit of reparative mechanisms, which is available to many other cells. What is meant by terminally differentiated cells? That is, they do not have any reparative mechanism. In case any damage happen to sperm, it cannot autocorrect it. So that's why that is what the meaning of terminally differentiated cells. Sperms sustain oxidative damage during the transport. Sperms are allogenic to female. They may encounter defenses of female immune system. The epididymis is divided into caput, corpus and cauda. We can see here this is the epididymis and this is the caput, means the head of the epididymis. Head of the epididymis is known as caput. Here there is beginning of sperm maturation, while this is the corpus. Corpus is where the maturation of the sperm continues. The body of the epididymis is known as corpus, while this is the cauda, the immediately down one, from where the vas deferens further starts. So this is the site of final maturation and storage of the sperms. So sperm storage happen in the cauda epididymis. The sperm reach cauda epididymis 72 days after initiation of the spermatogenesis. The cauda epididymis stores the sperm available for ejaculation. Preservation of optimal sperm function during the period of storage requires adequate testosterone levels in circulation and maintenance of normal scrotal temperature, which is usually 1.5 to 2 degree lesser than the normal body temperature. The spermatogonial cells are extremely heat sensitive. Any temperature more than 2 to 3 degrees centigrade then the scrotal temperature can lead to damage to the dark spermatogonia, which are the precursor for further spermatozoa formation, like the excessive use of disposable nappies and the excessive use of sauna, exercise, tight underwears. All these things can lead to the damage of the spermatogonia and ultimately resulting in azoospermia. Coming on to site of semen deposition, it is an anterior vagina near the cervical os. Events post ejaculation, semen coagulates within a minute of ejaculation and then liquefies within 30 to 60 minutes. The predominant structural proteins of the gel are semenogalin 1 and 2, which are secreted by seminal vesicles. The gel is degraded by prostate specific antigen. So the coagulum formation is because of the seminal vesicles, while the liquefaction is because of the prostate gland, because it releases prostate specific antigen. What is the role of that coagulum? It holds the sperm at cervical os, which acts as a copulatory plug and protects the sperm against a harsh environment of vagina, because vagina has a pH of around 4.5 that can lead to damage to the sperm. So that's why the coagulum has its own role. What are the vaginal defenses? First is the acidic pH 4.5. Second is the immunological responses. Mechanisms applied by sperm to combat the vaginal defenses, the immunological as well as acidic response of vagina. How the sperms combat with that? First is the semen is alkaline, so the ultimate pH which will form will be neutral. Inhibitors of immune response in seminal plasma also exists, inseminating many sperms, thus outnumbering the leukocytes. So because the male semen, it has a lot of spermatogonia, so which usually outnumber the leukocytes available in the vagina to kill the sperms. This protection is transient, and most sperms left in the vagina are immobilized within 2 hours. We can see here the sperm attrition. Sperm deposited in the vagina 200 to 300 million, while sperm achieving proximity to the egg only a few hundred. Now what are the causes? First is expulsion of semen from introitus, which we call as flowback, which is normal even in normal women. It happens, not only in infertility. Flowback is a very normal phenomenon. Second is digestion of sperm by the vaginal enzymes. Third is phagocytosis of the sperm along reproductive tract. Fourth, sperms burrowing or engulfed by the endometrial cells. Fifth, sperm lost into the peritoneal cavity. So these are causes of the sperm attrition. In spite of millions of sperms which are deposited in the anterior vagina, still only a few hundred reaches the fallopian tube. Sperm transport through cervical mucus. The sperm swim and migrate through the pores in the cervical mucus that are smaller than the sperm head. Therefore, sperm must actively push their way through the mucus. Cervical mucus has a filtering action. The abnormal and less capable sperm have difficulty getting through. Abnormal morphology of sperm head is associated with impaired flagellar function and poor mucus penetration. So basically, cervical mucus acts like a sieve. It acts like a sieve, and it only allows the good quality spermatozoa to further ascend into the female genital tract. We can see here, this is a picture of electron micrograph picture of the human cervix, and we can see a portion of the wall has been removed to reveal the architecture of the cervical canal. This CC means cervical canal, and these are the large primary grooves. We can see the PG is written, that is the large primary groove, can be seen at the external os that extend deep into the cervical canal. We can see here the secondary grooves. The smaller one here is the SG is written, it is the secondary grooves. Although the primary groove appear to form a preferential path for the sperm, it is not known whether secondary or even the tertiary grooves could and blindly and entrap the sperm. So are sperms stored in the cervix? Vigorously motile sperms have been recovered from the cervix up to 5 days after insemination. It is not known whether sperms collected from the cervix this long, means 5 days after the coitus, would reach fallopian tube and fertilize. Nor could it be known whether these sperms had re-entered the cervix from the uterus. So still it is not clear whether the sperms are stored in the cervix or they are just re-entering the cervix from the uterus. Sperm transport through the uterus, cranially directed waves of uterine smooth muscle contractions increase in intensity during late follicular phase. So the contractions, they become cervicofundal. So cervicofundal contractions help in ascent of the sperms. In periovulatory period, contractions limited to layer of myometrium directly beneath the endometrium. Contractions occurring during menses involve all layers of myometrium. Contractile activity of uterine muscle draws sperm and watery mid-cycle mucus from cervix into uterus. So uterine cavity is traversed in less than 10 minutes by sperm swimming at about 5 millimeters per minute. Sperm transport through uterotubal junction. The uterotubal junction presents anatomical, physiological, or mucous barrier to sperm passage. It traverses a thick muscular layer of uterine wall filled with viscous mucus, impeding progress of sperm. Sperm surface proteins required by each sperm to pass through the junction. One is fertilin beta, calmagin, and testis-specific angiotensin converting enzyme. These are the sperm-specific proteins which are required by each sperm to pass through the uterotubal junction. Normal morphology and motility are not sufficient for enabling sperm to pass through uterotubal junction. Sperm surface proteins are required by each sperm for them to pass through the uterotube. Fallopian tube, is it a sperm reservoir? As sperm pass through uterotubal junction and enter tubal isthmus, they may be trapped and held in a reservoir. Storage reservoir maintains fertility of sperm until ovulation. Poor sperm interaction with endosalpingeal epithelium might be associated with reduced fertility, such as that often observed in women with endometriosis. So, human sperm must be stored somewhere in female genital tract, and fallopian tubes are strong candidate for storage sites. We can see here the electron micrograph of the fallopian tubes. Here the arrows are marking the spermatozoa which are attached to the ciliated area of the fallopian tube epithelium. Advancement of sperm beyond uterotubal reservoir. Sperm undergo two changes in preparation for fertilization. The first one is the capacitation and the second one is the hyperactivation. Now, what is capacitation? Cellular changes ejaculated spermatozoa must undergo in order to fertilize. Capacitation occurs in fallopian tube. This is a very important point. It occurs in fallopian tube and is characterized by three accomplishments. First is the ability to undergo acrosome reaction, the ability to bind to zona pellucida, and the acquisition of hypermotility. Now, what is hypermotility? Rapid lateral oscillations of head about a fulcrum at head-tail junction, which we can see in the GIF which is being played. These movements, the lateral oscillations, they are at the head-tail junction, suggesting a kite-like action on the zona. Active motility of both head and tail are happening, as we can see very clearly. Hyperactivation is required by sperm to navigate toward oocyte and penetrate its investments. It assists sperm in detaching from endosalpingeal epithelium. It enhances ability of sperm to swim through viscoelastic substances like mucus in tubulomen and extracellular matrix of cumulus oviparous. Hyperactivation endows sperm with greater flexibility for turning around in pockets of mucosa. Hyperactivation assists sperm in navigating increasingly complex maze. In the fallopian tube, the mucosal fold increases in height and branches from isthmus to ampulla. And finally, it aids sperm in penetrating zona pellucida. Capacitation induced changes in sperm head surface are responsible for reducing binding affinity, and pull produced by hyperactivation can hasten detachment of bound sperm from epithelium. So both capacitation as well as hyperactivation are very important for natural fertilization to happen. Taxis of sperm toward oocyte. Two complementary guidance mechanisms operating within fallopian tube are: first is the thermotaxis. Temperature difference of up to 2 degrees between cooler tubal isthmus and warmer tubal ampulla has been detected. The capacitated sperm tend to swim toward the warmer temperatures. Second is the chemotaxis. Once in tubal ampulla and at the closer proximity to oocyte, chemotaxis guides sperm closer to the oocyte. Now, what is the fate of non-fertilizing sperm? They are either phagocytized by the isthmic epithelial cells or they are eliminated into the peritoneal cavity. Female reproductive tract is shown in this image, which is illustrating the stages of gamete transport. This is a nutshell of all the discussion which we have done. In the A image, we can see the sperm entering the cervical mucus at external os of the cervix. The mucus fills the upper half of the insect. We can see here in the box. B, the sperms interacting with endosalpingeal epithelium in the fallopian tube. C, the hyperactivated motility of sperms in the fallopian tube. D, the oocyte in cumulus within a transverse section of the tubal ampulla. So thank you everyone. I hope this was clear, because sperm transport is something which is a very less discussed topic. So that's why I tried to cover this in basics of reproductive medicine. Thank you.